Hello, I'm Tom Carmichael, Chair of Neurology at UCLA, and today I want to talk about National Institutes of Health, or NIH funding to biomedical research. There have been recent proposed widespread cuts to this funding. These cuts have been proposed in the Facilities and Administration, or F&A fees, for biomedical research. At UCLA, these are 57% on top of the original research grant, and there have been proposals to cut this to 15%. This will have substantial negative effects on the infrastructure that allows biomedical research to be done, but more on this later. First, it's worth noting that NIH funding is the main funding element for biomedical research in the US. It's the cornerstone of our fight to cure diseases. For example, in UCLA Neurology, NIH funding supports studies to understand cures and new treatments for Alzheimer's disease, Parkinson's disease, stroke, multiple sclerosis, and others. Without this funding, progress stalls, and so too stalls the efforts to transform human health. NIH funding and the F&A costs associated with it support research in laboratories, clinical trials, in epidemiologic studies which follow groups of patients with the disease over time, and in outcome studies which fund how disease treatments are working or not. As an example, in my lab, I study brain repair mechanisms in stroke and in dementia. We found new principles that might enhance repair and recovery in these diseases, and some of these have led to clinical trials. Let's go into the lab so that you can see how facilities and administration fees work in a biomedical research lab. Research funding in the US is principally from the National Institutes of Health, and this comes in two main forms. The first is funding for direct costs. These are the costs directly associated with the research that fund the salaries of the lab technicians and the trainees, the supplies, and a portion of the researcher's salary, and indirect costs, which support the infrastructure, the building, the facilities, and the administration costs that actually help the research get done. These indirect costs include heat, ventilation, and air conditioning, custodial services, electricity, gas, administrative oversight of chemicals and biological reagents, clinical trial oversight, purchasing and payroll administrative services, and network and IT infrastructure. All these buildings and facility support costs are called indirects, or F&A costs. With a substantial loss of this kind of funding, the building can no longer support the lab that gets the research done. Another way to look at this is to imagine that you got a large grant, a big chunk of money, to open a world-class pizza restaurant. The money comes and it funds the dough, the sauce, the toppings, the cheese, and you think, I'm going to make a world-class pizza. And then you realize the money doesn't fund the oven to bake the pizza, or pay for the rent so that you're making the pizza in the pizza store and not in the parking lot, or the electricity to keep the lights on, or the salary of the dishwasher who cleans the dirty glassware and plates, or the Wi-Fi that allows you to Google, why is my pizza dough not rising? These are essential costs for the restaurant, essential ways you make the pizza, but they're not directly funded by the grant that you got for the world-class pizza restaurant. These are the indirects. These are the costs for the facilities and administration of grants that allow the groundbreaking discoveries, the ability to fight disease and to study new cures and new therapies, the ability to keep the lab and the clinical trials running that are outside of the direct costs for those studies. Without the ability to have the oven that makes the pizza, the science stalls, and so too does the ability to move forward in biomedical research.